Hello, welcome to my channel, AI Medical School. So, today our topic is ischemic and hypoxic injury. Causes of hypoxic injury. Number 1. Ischemia-lack of blood supply due to obstruction of arterial blood flow. Number 2. Decreased oxygen carrying capacity of blood as in anemia and carbon monoxide poisoning. Number 3. Decreased oxygenation of blood due to respiratory diseases. And, number 4. Decreased tissue perfusion as in hypotension and shock. Features of reversible injury. Hypoxia affects mitochondria that results in decreased synthesis of ATP. As a consequence of reduced ATP availability, following changes develop in cellular structure which reversible if oxygen supply is restored. Number 1. Cellular swelling. Number 2. Desegregation of ribosomes and failure of protein synthesis. Number 3. Reduced intracellular pH resulting in clumping of nuclear chromatin. And, number 4. Appearance of myelin figures and cell blebs due to membrane damage. Let's discuss it little bit in detail. Cellular swelling. Cellular swelling or cloudy swelling, or hydropic changes characterized by the presence of large vacuoles in the cytoplasm. Cellular swelling is the result of failure of cell membrane pump which maintains sodium-potassium balance across the cell membrane. Failure of this sodium-potassium ATPase pump due to deficiency of ATP results in accumulation of sodium inside and potassium outside the cell. Excess sodium inside the cell exerts osmotic pressure and pulls water inside, resulting in cellular swelling. It is the first manifestation of all forms of injury to cell in which cell becomes incapable of maintaining ionic and fluid homeostasis, resulting in cellular swelling, pale color, and increased weight of the organ. Desegregation of ribosomes and failure of protein synthesis. Ribosomes become detached from the rough endoplasmic reticulum due to its swelling, and therefore protein synthesis is reduced. Reduced intracellular pH. Decreased ATP synthesis stimulates the enzyme phosphofructokinase activity, resulting in increased rate of anaerobic glycolysis that leads to production of pyruvic acid and subsequently lactic acid. Accumulation of lactic acid decreases intracellular pH, which causes clumping of cytoplasmic organelles. Disruption of lysosomal membrane leads to release of lysosomal enzymes into the cytoplasm, which damage vital intracellular molecules. Appearance of myelin figures and cell blebs. Myelin figures, intracellular wall-like structures originating from damaged membrane. Cell blebs, a cell membrane deformity most likely caused by disorderly function of the cellular cytoskeleton. Features of irreversible injury. Nuclear changes. Nucleus may show one of the three patterns of changes. Karyorrhexis. The pycnotic nucleus may break into numerous small particles. This process is called karyorrhexis. Karyolysis. When the nucleus undergoes lysis without pycnosis, the process is called karyolysis. Cytoplasmic changes. Irreversible damage to mitochondria manifested by severe vacuolization. Extensive damage to the plasma membrane. Massive calcium influx acting as a poison for mitochondria. Loss of enzymes and proteins due to increased membrane permeability. Lysosomal swelling and leakage of enzymes. Last point. Injury to the lysosomal membranes results in leakage of their enzymes into the cytoplasm, resulting in enzymatic digestion of cell components, autolysis. Leakage of these enzymes into circulation after cell death provides an important means of detecting tissue-specific cellular injury and death in blood serum samples. For example, cardiac muscles contain specific enzyme creatinine kinase. In myocardial infarction, serum level of this enzyme is raised, providing a good indicator of myocardial infarction. That's it for now. I hope you like it. Thanks for watching, and don't forget to press subscribe button.